As last speaker of this webinar, my talk is going to be focused on advanced organoid culture systems and data applications in infectious diseases. During my talk, I would like to give you first an overview on how organoids can be used as a tool to perform studies of infectious diseases, and then focus on the second part on what tools are available at STEMCELL to assist scientists in studying SARS-CoV-2. Let's start. Organoids are defined as a collection of organ-specific cell types that develops from stem cell or organ progenitors and self-organizes through cell sorting and spatially restricted lineage commitment in a manner similar to in vivo. As described by this definition, organoids, as for example intestinal organoids, can be generated from crypts isolated from mouse models or patients, and be also generated from human embryonic stem cell or human induced pluripotent stem cell directly differentiated into organoids in vitro, or by reconstituting organoids derived from biobanks. Organoid in vitro cultures could be used for multiple downstream applications, such as high-throughput drug screening, genetic profiling, host microbe interaction. By the use of genetic editing, such as CRISPR-Cas9, organoids can be used to perform targeted mutations and study disease models. Organoids are used a lot today for generating organoids on a chip and for creating in vivo transplant models. As I said, this presentation will be mainly focused on the host microbe interaction. And for this reason, on top of what has been already widely discussed by previous speakers of this webinar, I would like to highlight some characteristics of these culture systems that are really important for studies that focus on infectious diseases. Organoids are 3D cultures in which the lumen of the epithelium, as you see here, is found enclosed within the structure itself, within the epithelium. And these allow very limited access to the apical side of the epithelium, which is usually the interactive surface. This aspect of this culture system is really critical when we come to talk about infectious disease studies, as I said before. Acknowledging this structural impediment, scientists established different ways to be able to get access to the apical side for performing infection studies. There are different approaches, and one is through the fermentation of the organoids and the incubation of the fragments with the microbes for a certain amount of time, to then reconstitute the organoids in an in vitro system that is, again, matrigel based, and be able to analyze secondary effects of this infection in time. Another approach that is very effective in studying the interaction of the pathogens with the epithelium consists in directly inject the pathogens within the lumen of the organoid. Finally, if researchers need to study viruses targeting specifically fully matured differentiated cells, they can definitely fragment the organoids, seed these cells from standard plate or transfer system, and generate a nice monolayer on top of which microbes can be applied to the apical side. So using these approaches, the researchers managed to model infectious diseases of pathogens infecting human beings that are generally poorly replicating in transformed cell lines, and due to their host range restriction, they are unable to replicate in many animal models. Now, I would like to give you some examples of studies that make use of organoids to establish infectious disease modeling. In the first paper, I would like to review with you scientists from the lab of Hans Clevers established culture conditions for maintaining gastric organoids in vitro. In the same paper, they utilized these organoids to put in culture a pathogen that was not possible to culture in vitro before. Microinjection of the Helicobacter pylori straight into the lumen of the gastric organoids allow the culture of this pathogen that, as you can see here, remains very limited and confined within the organoid. And they could also observe a nice interaction with the epithelium at the level of the electronic microscopy. In a follow-up paper, scientists use gastric organoids to model the interaction not only of the Helicobacter pylori with the epithelium, but also with immune cells. In this paper, very nicely, they inoculated dendritic cells within the culture system, specifically in the medium, and they observed migration of the dendritic cells that localize mainly around the gastric organoids that are infected with Helicobacter pylori. And they very nicely show how dendritic cells are able to perform phagocytosis in vitro. Another example of modeling virus infection in vitro comes from kidney models. Scientists managed to establish tubuloids culture derived from human adult kidney and urine, and use this culture system to model the infection of BK virus, which has never been performed before. Microinjection of the tubuloids with the virus allowed the scientists to observe the effects that can recapitulate the nephropathy happening in vivo. They observed enlargement of the nuclei on the perimeter of the kidney organoid epithelium. These nuclei seems to be positive for viral protein. They could observe many viral particles within the cells in culture and also model the replication of the virus in this system. But very importantly, they show how this culture system could be used for drug screening purposes, as you can see here. Intestinal organoids have been also used to better understand targeting mechanisms of different viruses, such as rotavirus. After establishment of an efficient infection system, culture system, as you can see here, they managed to show a high resolution that rotavirus is able to target specifically enterocytes and endocrine cells. Intestinal organoids have been also used to better understand how environmental factors can influence viral replication of norovirus specifically. In this publication, they took advantage of intestinal organoids culture system, generating intestinal monolayers in which they inoculated norovirus. As they show here, the virus nicely infects the epithelium and replicate in vitro. But very importantly, they could observe how bile acid content in the medium, in the system, is actually influencing different genetic strains of this virus. These are observations that could never been done before. These papers that I summarized, I believe, gave you some examples on how scientists use organoids to characterize key mechanisms that were impossible to be observed before at this so high resolution level. Specifically, the establishment of monolayers really reminds me about an established model system that is used for studying respiratory infection disease. And as you may know, I'm talking about air-liquid interface type of cultures. Air-liquid interface is considered the gold standard for respiratory infectious disease studies. As you know, air-liquid interface cultures are model systems based on the generation of monolayers, as you can see in this cartoon. In this culture system, the virus can be inoculated on top of the monolayer generated in the insert of the transfer. And many different parameters can be studied at this point. People can look at the top of the monolayer and get information about mucociliarity clearance, cilia beating frequency, perform an ELISA for apical cytokine analysis, perform tear measurements, and morphological evaluation. Scientists can also get information about the cells, looking at intracellular viruses, viral spreading, characterization by histology or ICC, perform ion channel activity assays, or study signaling pathways. Other information can be also retrieved from the basolateral side of the culture system by looking at cytokine release and RDH release. I'm going to give you some examples on how primary airway air-liquid interface cultures have been used to characterize molecular mechanisms of respiratory viruses. And I will start with the respiratory syncytial virus that, thanks to the air-liquid interface, has been shown to target specifically ciliated cells. As you can see, these monolayers have been established in an air-liquid interface culture system, and scientists show that in this very nice polarized epithelia, RSV-tagged GFP virus is able to infect specifically the ciliated cells. Air-liquid interfaces have been also used to model how rhinovirus attacks the lung epithelium. And it's shown here that rhinovirus specifically targets the ciliated cells, but also upon infection of the cells, rhinovirus causes loss of the cilia. As you know, at the moment we are fighting a very big war against a specific respiratory virus. And you can imagine that I'm referring to severe acute respiratory syndrome, SARS-CoV-2 virus. So coronaviruses have been studied for a really long time now. The first observation of a coronavirus occurred in 1975 when scientists described a virus-like particles isolated from human feces. In 1994, another group classified these virus-like particles being part of the Coronaviridae family, again isolating them from stool samples of affected patients. Scientists have also used intestinal organoids previously to model coronavirus infection. In 2017, this group studied how MERS, another coronavirus that had its outbreak in 2012, interacts in the intestinal epithelium. They show that the virus infects the intestinal epithelium at the intestinal biopsies by looking at the nuclear protein, and then they establish replication of the virus infecting intestinoids. That's the way they call it in this paper. So they can see replication of the virus, and they can characterize the infection by looking at fixed sections of these organoids. As you know, last December, the first outbreak of SARS-CoV-2 occurred in Wuhan, China. And World Health Organization declared COVID-19 a pandemic on the 11th of March 2020. One common way of transmission of SARS-CoV-2 is aerosolized particles. Lungs seems to be the primary site of infection from where the virus might spread in other organs. The incubation period is approximately between 3 and 14 days. Although it seems that lungs are the most affected and targeted epithelia by the virus, it's becoming clear that perhaps the effect on other epithelia have been underreported and maybe need further investigation. Something to think about is that besides the common symptoms that are indicated by the World Health Organization, being fever, dry cough, and fatigue, there are reports that indicate other common symptoms affecting multi-organs, such as, for instance, the intestine, as other coronavirus have been already described for, or in severe diseases, also affecting the kidneys. As you know, at STEMCELL, our motto is scientists helping scientists. And I would say that we are trying to help scientists in this moment fighting COVID-19. I would like now to talk about some tools already available or under development that could be used for study this virus from different point of views. They can go from basic research, understanding basic molecular mechanisms of infection, which we really need because we don't have so many information about this virus. And they can span to the level of applied science and drug screening and drug toxicity assays. As a first tool provided by STEMCELL, I would like to introduce the pneumocult workflow that allows differentiation of the human primary bronchial epithelial cells in vitro on a transwell insert. Air-liquid interfaces can be differentiated using our media, and the cells show full differentiation of goblet mucus-producing cells here, and formation of nice tight junctions and formation of cilia on the apical side of the monolayers. Another system that is under development, and it will be soon available, consists in the differentiation of the human primary bronchial epithelial cells into 3D matrigel-based system forming organoids. These organoids show the differentiation of cilia that protrude to the apical side of the epithelia. And they're also able to differentiate goblet cells. These stainings show really well how this platform really mimic in vivo systems. As I mentioned in the first part of this presentation, air-liquid interface systems are the gold standard for studying respiratory viruses. Knowing this, scientists working at the CDC in China decided to use this culture system to identify what was causing this terrible bilateral pneumonia that was leading to death of so many people. The severity of the situation really pushed these scientists to use a robust system for which they could obtain the result as quickly as possible. For this reason, they decided to establish air liquid cultures using airway cells derived from specimens of surgical patients. Once obtained, the monolayers formed, as you can see in this cartoon, they applied bronchoalveolar lavage fluid derived from the patients presenting severe pneumonia to the apical side of the epithelial cultures. Using a liquid interface, after only a few weeks from the outbreak, they managed to identify SARS-CoV-2, sequence its genome, and make these data available to everybody else in the world in order to start fighting this very dangerous pathogen. Since then, the characterization of the virus is continuing all around the world. And something that people are really focused on at the moment is to understand how SARS is able to infect the cells. It has been shown that SARS-CoV-2 is very genetically similar to SARS-CoV, another coronavirus which had its outbreak in 2002. And similarly to SARS-CoV, SARS-CoV-2 specifically interacts with angiotensin converting enzyme 2, called AC2 receptor, that is expressed not only, unfortunately for us, by the pulmonary tissue, but also by other tissues such as heart, kidney, endothelium, and intestine. Really, because of these multiple organ expression of this receptor, people observe multi-organ dysfunction in patients. So knowing this, we ask the question whether human airway epithelial cells cultured in pneumococcs express this receptor, and whether then this system could be a good model for studying SARS-CoV-2. As you can see here from these very nice stainings, we can observe polarized epithelial monolayers that express high levels of this protein on the apical side, both for bronchial airway epithelial cells and human small airway epithelial cells, suggesting that this system could be a potential platform for studying SARS-CoV-2. Many studies and analysis of cases around the world suggest that intestine might be a major entry site for SARS-CoV-2. It has been reported that patients tested negative for the nasal swab come out positive for the presence of SARS-CoV-2 in their stool, unfortunately. And as you know, at STEMCELL, we have a nice workflow for generating human intestinal organoids. We characterize the organoids grown in testicle OGM, and we show by the staining, positive staining for Ki67, that these organoids present cells that localize mostly on the crypt region of the intestinal epithelium. We ask the question whether the organoids can express AC2 on the apical side. So we perform the staining and we observe a low expression of AC2 on the organoids cultured in intestinal OGM. As you can see from this staining, only specific cells are expressing a low level of AC2, suggesting that these cells might be progenitors of enterocytes or immature enterocytes that need to be further differentiated in order to maybe express higher levels of this receptor. So we know that AC2 is very highly expressed indeed in the villi region of the intestinal epithelium. And we asked the question whether we could establish culture conditions to trigger the differentiation and the maturation of this progenitor into more matured enterocytes in order to obtain AC2 expression levels similar to in vivo. To do this, we used our intestinal organoid differentiation medium workflow that allows differentiation of organoids generated from human biopsies of the intestine or other organoids that can be passaged and maintained long-term in culture. It can be also differentiated in the 3D type of culture systems, keeping the organoid shape, or they can be passaged and generated monolayer in a standard plates or transwells that can be airlifted to generate finally air-liquid interfaces. In these systems, we characterize the expression of enterocyte-specific markers, such as vilin, and goblet cell-specific markers, such as mucin-2, in both organoids and monolayers type of cultures. So we thought to test now the expression of AC2 on these platforms. And we show here a very nice apical expression of AC2 receptor gaining on the epithelial generated in the submerged and air-liquid interface culture systems, really suggesting that this could be another platform useful for further research. Another possibility to research for COVID-19 is represented by kidney. Because as I said, kidney results to be failing in severe cases. So AC2 receptor is very highly expressed in the tubules of the kidney and lowly expressed in the glomerulus, as you can see from this picture taken from the Human Protein Atlas. A recent paper that is in press right now used human pluripotent stem cell derived kidney organoids as a platform to model the infection of SARS-CoV-2 in vitro. They perform single-cell RNA-seq analysis, and they show that AC2 very nicely localized within the proximal tubule and the podocyte type 2 cells. On top of this, in the same paper, they validate this platform to be used for drug screenings. Specifically, they tested human recombinant soluble AC2 and show that is able to stop the proliferation of the virus in the kidney organoids, as you can see here. Now, at STEMCELL, we have been developed a kidney organoid platform to differentiate kidney organoids directly from human pluripotent stem cell lines. This protocol has been optimized from the technology published by Joseph Bonventer and Benjamin Friedman. And we show that our organoids are able to differentiate podocytes, proximal tubules, and distal tubules. Organoids derived with the STEMDiff kidney organoid kit are compatible with high throughput formats. As you can see here, we show very nice picture of a 96 well plate in which kidney organoids have been differentiated from 3 different cell lines. And you can appreciate the number of different organoids generated per well, plus the endothelial cells that are surrounding the specific structure of the kidney cells. So the kidney organoids generated using STEMDiff kidney organoid kits are composed, as I showed you, by proximal tubules and distal tubules, which are the main AC2 expressing cell types. The HPC-derived kidney organoids that were being generated by Nuria Montserrat in Penninger Cell paper They show to express AC2 at very nice levels, and they can be utilized to perform studies on SARS-CoV-2. Now, our team is very much focused in confirming the expression of AC2 receptor and its regional specificity at the gene and protein level in our system. I showed you some of the tools that STEMCELL can provide for supporting research against COVID-19. We hope that all these tools can assist in better understanding the nature of this virus and how it targets the different tissue types and potentially be used as platform for drug discovery. At the end of my presentation, I would like to share with you a message from my little princess. Similar to many children around the world, she's spending her time these days enjoying nice rainbows, as you can see here. And she's sending her own message to the world saying that everything is going to be fine. Andrà tutto bene. I think that this will be possible thanks to everybody's contribution. In particular, I would like to take the chance to thank all the frontline workers who are the real fighters of this war. We'll manage to re-emerge from this really difficult moment of history, stronger and more united than we were before, only thanks to their daily effort. Thank you all again, and I will be happy to take any question. Thank you.